it out.
Sure. Well, after I get that finished, and apparently now I have a deadline for her.
No, because of the door structure at Shapiro, that's an easier thing to enforce. It's not like you can get into that floor any other way than through the front entrance and then there's glass doors. So we'll just exit through Hatcher, it'll be easier.
It's really hard for me to play like that though. I feel like I block like the other.
Then I just have to worry about the huge pavilions that they've built all the way, you know, between
No, I mean, like, I first started to play
the crowd and Rackham. Don't want them for, yeah, the expo. Yes, we have that house. I'm girding myself for it already.
Can you just skip tomorrow? Go work somewhere else?
So I don't think actually. But no, I think have you been saved from the insanity that
Don't tempt me.
It looks like a yes. I don't think I've told her about this at all. Actually.
Right.
I just don't know if you can be. We have a whole conversation. Love life. It might be. You're now into a cup.
And since you guys have a coffee shop, I mean,
That's the first time we've seen the Rockies. Or motion picture or whatever for like 5 years. What is it?
Like here or
Or a picture show? Like I had a bunch that I was looking at. I started looking at one day. They're like pretty decent prices.
When's it look
I literally told one on Monday. My sister's getting married in December.
so can I ask a kind of ignorant question? When did the announcement go out that Lynn was leaving Tubman?
Were you get email from Marissa at some point to say like she's gone?
She's not going to be on the, like she's not going to be on the data education working group.
Okay, so no one else would know. Basically, I saw an email one day.
I didn't miss anything. Okay, that.
I saw an email one day that basically said she's not here anymore. Please remove her from any email list that she's on.
Okay.
So
It's
yeah.
going. I don't know. I think it's going well.
Okay,
We've got
that
it
makes me feel
better now that everything's working.
less oblivious. So I found out because Amy Neeser had apparently been
2
talking
exams
to her.
and then a semester.
Now that Amy's in California, they had been talking about something. And Amy mentioned it to me, and I was like, "Wait, what?" She's like, "You didn't know?" But I'm like, "No."
Okay.
It's that first time, so it's gonna go, but they'll get something more out of it than.
Based on when.
But there was no.
How many are there?
Right. Okay. Okay. That makes.
Yeah.
That makes me feel simultaneously better and also about it. We don't leave it at that. It's more one.
So, for the rain.
So we're done now. 5 weekends in a row. Like you're being your GSI. That's like something you put in the positive.
What day you get
Okay.
That's one thing we're like, oh, what do you want like it's kind of like for like I don't get it $10 for editing writing. Am I right? It takes like a few hours, so that's why I was probably going to do it. Like it was, I could drive, but then we would have to leave to drive back on the second from Kansas City. So it's probably trying to figure out
probably
that's right.
Clean the mics.
It's around, so.
Sure. All right. Sorry. Alright, welcome everyone. Tools and Technology Seminar Series. I just started passing around the sign-in sheet. If you haven't signed it already, please do so when it comes around. If it never gets to you, feel free to sign it before you leave today. So today's speaker is Golam Mortuza, and he is a postdoc in Yang Zhang's lab in DCMB.
Thank you so much, Marcy, for the introduction. So he already mentioned, like, I'm the postdoc from Yang Zhang lab. You can call me by Golam. So I'd like to thank Marcy, first of all, to invite me in this seminar series. So today I'm going to talk about the ab initio structure prediction of proteins, where we use the contact information between the residues of the protein. So that's why the title of my talk is Contact Assisted Ab Initio Structure Prediction of Proteins. So the outline of my talk will be, first I'm going to give you a brief background, how protein structure can be helpful to understand protein functions. And then I'm going to talk about the pipeline that we have in our lab, Quark, which is used to predict structure of protein using ab initio technique. And then I'm going to talk about the next pipeline that recently is developed in our lab called NEPCON, which is used for predicting contacts of residues in the proteins. And finally, I'm going to talk about the combination of NEPCON and Quark. We call it C-Quark. So it's basically contact-guided ab initio structure prediction program. So as you all may know that protein is a biological molecule which performs essential cellular functions or in the organism, like cell transporting and also like carrying oxygen to the blood, cell signaling, and so on. So these biological functions greatly depend on the structure of the protein and their sequences. So let me give you one example where I'd like to illustrate this point, that how protein structure can be helpful in understanding protein-protein interaction. So this work was done by Dr. Yang Chao. He's from Sichuan University. So whenever he did this task, he was in China, but then after that, he actually came to our lab as a visiting scholar. So here, basically, this pink protein is MYC6, which inhibits cell migration by binding with this blue protein called CDC42. So we actually want to know that what are the hotspot of these MYC6, or basically what are the key residues or amino acids that bond with this blue protein CDC42 that inhibits the cell migration. Now we have a solved structure, or experimentally solved structure of this blue protein, that's CDC42. But the domain that interacts of domains of MYC6 that interact or bond with this CDC42, we don't have any structure for that. So what we did, we predict a structure model of this MYC6 using iTaser. You might have heard about it, and I'm going to talk about this also later. So we predict the model, and then we dock this MYC6 with this blue protein using ZDock, and then find out the hotspot using server which is used for like a hotspot identification. I think it's KFC2. So then after that, we find out that there are 4 residues in this MYC6, that I11 isoleucine, R30. So this number is basically the index of these residues in the protein. So arginine, methionine, and also again arginine. So these are the 4 residues which basically are key residues which bind with this protein and inhibits the cell migration. So this is a predicted model. So in order to test that, we also had a wet lab experiment where the top panel is basically the CDC42, this blue protein, and in the middle panel, if you see any black strip, that means there is an interaction. So this is the wild type. So for wild type, you can see that CDC42 and MYC6 has a good interaction. As I mentioned, that these are the 4 key residues which bond this MYC6 with the CDC42. So what we did, we actually muted these 4 residues to alanine. Like here in this column, we muted this methionine or arginine to alanine. So here you can see this black strip actually become weaker. That means the interaction is getting weaker, and here again the isoleucine and arginine also mutated to alanine, and it became again weaker. And in this column, we actually muted all of these key residues to alanine, and there is no interaction at all. So this experiment actually shows that how our model can be helpful to understand the protein-protein interaction. Okay, so even though, you can see that structure prediction is helpful for protein function inference, but unfortunately, the number of solved structure of protein actually lags far behind so far than the number of sequences or protein sequences so far discovered. So I took this data from UniProt database yesterday. So here this plot actually shows in different years how many number of sequences of proteins are deposited. So these are only sequences. And here as of 2017, it's actually approximately 90 million. Okay, and you can see there is a peak down because there are some redundancies, so they removed that, and that's why it again dropped back. So this is currently the 90 million is protein sequences discovered, but among these 90 million, only 120,000 of that, approximately 120,000 of that, actually have solved structure which is deposited in the protein data bank or PDB library. So you can see the gap is like 775 times, and it's only 0.13% of the total number of proteins. So there is a huge gap, right? So we have to minimize this gap. So that's why our research is to predict accurately or model the structure of protein, so that we can, with the hope that we can reduce this gap first of all, and also these models can be helpful to understand the protein functions and for biological applications like drug discovery and so on. So we have several pipelines in our lab. One of them you might heard, iTaser, I guess. This is actually a very popular program in our in this research community. So just to give you some data, so we have more than 83,000 registered users to use this program across, I think, over 130 countries. I think it's right now 132. So you can see it's very popular, and it's getting popular and popular due to the accuracy of this prediction. So let me walk you through the pipeline of this popular program. So here's the sequence, and we want to get this structure, right? So it starts from sequence. So what it does, so it starts these sequences through the protein data bank, so where we have all the experimentally solved structure. So it starts through that and try to find out that potential structural homologous protein that is kind of structurally homologous to this sequence. So we call it template. So we use our in-house program called LOMETS threading program, which does that to find out this template. So this template is then is assembled in the structural assembly simulation using Monte Carlo, and then during this Monte Carlo simulation, basically we have more than thousands of conformation that basically starts from a random configuration and tries to go to the energy minima to get the correct fold that we look for. Now during this, as I said, there are more than thousands of conformation. We call it decoys. So what we do, we actually cluster those decoys based on their structural similarity. And these decoys are again then used as a restraint for the next round of simulation. And then we do some refinement finally to get the structure. So what I mean by refinement, so in order to accelerate this simulation process here in the Monte Carlo, we don't use the all atoms. We just use like the backbone atoms, okay? But the final structure should be full atomic. So that's why finally we do some refinement to get the final structure, okay? So this is the overall pipeline. So here one thing you can see, the accuracy of this model greatly depends on the template, right? So if we don't a good template hit, which is like structurally, which might be potentially like structurally homologous to the sequence, then there might be a chance that the accuracy of this prediction is low, right? So that's kind of one of the limitation, and that's why we call it template-based structure prediction, because it uses templates. So to give you some perspective, so we actually tested this method on using like 489 proteins, which has already solved its structure, okay, to benchmark. So after we get the model using iTaser, we have to assess the model, how good it is, right? So what we do, we superimpose this model with the native structure that has the already solved structure, and then calculate the RMSD, root mean square displacement. And if the RMSD is low, that means the model is pretty accurate or close to the native. So here I'm actually showing that out of these 489 proteins, the number of proteins that falls into a difference RMSD to native. So usually if the RMSD to native is in this region, like less than 2 to 2.5 angstrom, these models are really pretty good, and you can use this model for drug design or drug screening. Even if it is like less than 4 angstrom, you can use this for like docking or molecular replacement. And if it is less than 6 or 6.5 angstrom, then also you can use this model for defining the active catalyst site. But beyond 6.5, the model is not really very good. So here you can see that even if we would like to use this model for different biological function, out of 989 actually, like 66% of the time, ITER was able to predict a good model which can be used for biological function or biological application. The rest 34% time, it actually cannot predict a good model, and because it doesn't find a good template hit, and that's the limitation of this program or method. So for those 34%, we call it hard target, that we didn't find a good template. For those target, ab initio structure prediction can be helpful to correctly model those structure and then reduce the gap. So in our lab, we also have a program called Quark, which is used for ab initio structure prediction of proteins. So again, the pipeline actually looks like this. So it starts again from query sequence. Now, instead of getting template, what we do, we actually get a fragment, small fragments, like the range of this length is 1 amino acids to 20 amino acids. So for each position of the query, we actually find out that what is the good match or good fragment for each position. And then these fragments actually we randomly connect with each other and make a full size of protein, and then put that in the Monte Carlo framework. So this fragment generation has two tasks. One is, again, as I said, we get the fragment and then connect together and get this like initial configuration or conformation of this Monte Carlo simulation. This fragment also used to get the distance between these two residues in this amino acid. We call it distance profile, and I'm going to talk about this more details about this in the next slide. But the overall idea is like, so again, to guide our Monte Carlo simulation, we use this distance profile as a restraint, and also we have some knowledge-based statistical potential and also some physical potential as well to guide this simulation, where we have 9 types of movements to fold this protein, and we use replication Monte Carlo simulation for this, and then again we do clustering these decoys, do some refinement, and get the final model. Okay, so this is the overall pipeline of Quark that we use for ab initio structure prediction program. Now let me talk about this part that I mentioned, like how we generate fragments and how we get this distance profile, which is a valuable information to guide our simulation. So here, say this is the query sequence I have, okay? And as I said, in each position, we actually generate fragments which ranges from 1 amino acids to 20 amino acids. So the way we generate the fragment, so we align this each of this fragment of the query sequence with the template in the PDB or protein data bank, again the solved structure. And then we actually give a score to this alignment, and this is the scoring function where n is the residues in the query sequence and n is the residues in the template sequence. So we actually match their sequence profile and also their secondary structure. That means secondary structure between the query residue and the template residue, their solvent accessibility, their structure profile, and their torsional angle. So using this scoring function, we actually can rank all the fragments that we generated here to the descending order. And we only take top 200 fragments based on their scoring. Okay, so for each of these positions, we create top 200 fragments of this query sequence, if that makes sense. And then again, as I mentioned, we actually get a valuable information that what could be the distance, especially in three-dimensional, between these two residue pairs. Okay, because that's a very valuable information to guide your simulation. Now, how do you do that? So, say this is a fragment we find out from this in this position I, and this is the fragment we get from this position. So we actually check what are the fragment for each residue pair are from the same template PDB. What I mean is that, say this residue and this residue, or sorry, this fragment and this fragment at the two different position, if they are from the same template, then there is a high possibility that the distance between these two residues in the template should be the same distance of these two residues in the query, because these two residues should maintain the local environment in this the same environment for the same for different proteins. So that's an assumption. So we actually then check for a specific residue pairs I and J. We find out that, okay, what are the fragments are from the same template, and if we find out they are from the same template, then we find out their distances in the template. Okay, and based on that, we actually plot a histogram of the distance for each residue pair. Again, this is for only one residue pair, I and J I'm talking about. Okay, so we actually plot a histogram of distances for that specific residue pair, where we see that how many fragment pairs falls into different distances, and here these distances along in angstrom. Okay, so here I'm showing two representative histogram we can have from this data. One is that you can see that the number of fragment pair is basically increases monotonically with the distances, and we are not very interested in that, because this basically is due to the trivial increase of the distance. But we also see another kind of plot which is something like that. So we are more interested into this plot, which actually gets a peak. This plot actually means even though we gathered this fragment independently from different proteins or from different templates, but still for that specific residue pair I and J, there are most of the fragment actually falls into this distance, that means 6 angstrom for this specific case. So there is a high probability that this residue pair I and J can have this distance. So for each of this residue pair, we actually do this, like we plot this histogram, and then if we find any peak, we actually use this information in the Monte Carlo simulation, and we call it distance profile. So that's actually a very good restraint which can guide our simulation. So this distance profile is also combined with 10 other energy terms in the Monte Carlo simulation, where the first 4 terms we call it atomic level, like backbone atom or side chain atom. Then the next 3 terms is basically residue level, like solvent accessibility, torsional potential, and also the last 4 is topology level. And the distance profile actually falls into this residue level criteria. So using this energy potential, we actually then guide our Monte Carlo simulation, right? So this actually shows energy landscape for the protein folding simulation, and you can see it's pretty rough. So along x-axis, if you think x-axis, these are different conformation, and along y-axis, this is that energy. So there is a very good chance that if the energy barrier is too high, that your conformation might be trapped in the local energy minima, and if you are trapped in here, then you might not be able to reach to that energy minima we are looking for, because basically this is the native structure, and native structure usually has the lowest energy, so we always want to shoot for here. So to overcome this issue, we actually use a technique called replica exchange Monte Carlo. So what it does, so we start the initial conformation in different replicas, where each replica are in different temperature. For instance, in this example, I have 3 replicas, where the first replica is in temperature T1, T2, and T3. Okay. So in each replica, it actually follows a regular Metropolis Monte Carlo criteria. What I mean by that, so say you start an initial configuration or conformation, and then you give a random movement, okay, and then you calculate the energy compared with the previous time step, and accept the move based on this probability, and you do the same thing for each of these replica. We also compare the energy between the adjacent replicas. And then we can also move the conformation from one replica to another replica using this probability where the conformation from ith replica moves from ith replica to jth replica. Okay, and here again, as I said, that we actually now use different temperature, so that's why we can have, like, we can access different energies and which can actually follow or like guide our simulation to energy minima. And we have an energy distribution using this equation, which actually depends on the length of the protein, right? Because if you have bigger length, then you need more perturbation or more like higher energy to guide your simulation. So that's why this replication Monte Carlo technique helped us a lot to get the correct fold. Okay. Now again, after this Monte Carlo simulation, as I said, we do clustering and refinement and get the final model, right? So now how we assess our method is doing well, right? How we can assess that our model that we develop, it actually is close to native or accurate. So there are several ways to do that. One is you test on a lot of benchmark, like a lot of proteins, and then do benchmark, right? That's what we do as well. There is another way for us actually to assess is to participate in a CASP experiment. So CASP is basically critical assessment of protein structure prediction. So it's kind of like Olympic, for like structure prediction program. So basically this competition is held in every 2 years. So organizer releases like approximately 100 to 150 proteins which doesn't have any solid structure. Okay, and the goal of this competition is to assess the state of the art techniques in this research field. Okay, so the different groups across the world actually participate in this competition, and this actually goes like for 4 months. So they release like say 2 targets each day, and then we have to submit these like models in 3 days, okay, or in 72 hours. So after we get the give the model, so they already have the they also give this to experimentalists to solve the structure at the same time, and then to assess the model, they actually like give a DDT score, which range from 0 to 100. So higher the score, the better the model. So here I'm actually listing those servers which actually like in the like top 10 based on their accumulated DDT score. So the first one, the Zhang server, is basically our iTaser. We actually participate as iTaser. So so we actually participate like iTaser participate from 2006. So basically from CASP 7, and the CASP actually started from 1994. But Quark was developed in 2010, so we actually participated from CASP 9. So here you can see the first 2 groups or 2 server is basically our group or our program, like Zhang server and Quark, which actually like was the best method so far, And if you see this CASP 11 actually like 2014, I didn't show results for the last CASP that we participated. I'm going to actually show this later. Okay. So here, as you can see, that our model actually was again so far the state of the art, and it performed really well, our method. Now let me show you one example where the Quark really performed pretty well and very well accepted in this CASP community or CASP organizers. So this is the model that we developed, and this was like is released in CASP 11. It has 128 amino acids. So this is the model, and whenever they solve this structure, this is the crystal structure. Okay, so whenever we superimpose on each other, then the RMSD I already mentioned is close to 3 angstrom. That means the model is pretty good and close to native. We also have another evaluation criteria, we call it TN score, which is developed actually in our lab. So this TN score basically range from 0 to 1. So higher the value, the better the model is. And we actually find out that if your TN score is, if it is greater than 0.5, that means the model that you develop, it actually got the correct fold as the native. So here you can see the TN score is pretty high, and we actually got a correct fold. By the way, this actually picture you saw in my first slide, I guess, if you remember that. Now, whenever we use iTaser to model this, we actually get a TM score of 0.428, which is less than 0.5. That means iTaser was not able to get a correct fold for this, while Quark could. And the reason is that the fragment duty generated, those actually the average RMSD is like approximately 2 angstrom, which was pretty close to the native, and that really helped a lot. And the assessor's comment was that this target represents with biggest improvement for the PDB or protein data bank templates in CASP 11 experiment. Now you might be surprised, okay, Quark performs really well, better than iTaser, but in the previous slide I showed you Zhang server is the top, Quark is the second. Why is that, right? So there is a reason for that. So let me show you another example here, which was released in CAS 12, basically last year that we participated. So this is the native structure. So whenever we search through this, search the structure or the sequence through the PDB using low merge threading, the template we get, the TM score is 0.3. So as I mentioned, if the TM score is less than 0.5, the model is pretty bad. But this is the template. So you can see it's a pretty bad template. Now, if I use this template for iTaser, it's not going to work. It's not going to be able to get a good prediction. So what do we do? If we find out any target is very hard, that doesn't have good template, we actually model using Quark, because Quark can handle hard type of protein. And now you can see the TM score is pretty high, 0.49. And then we can use this as a template in our iTaser program, and then we can increase this TM score pretty high, because iTaser actually, the energy potential we have, it's pretty robust. It has more than 20 terms. So that's why whenever we use this template, use this model as a template, iTaser actually improve the model further, and that's why you see Zhang server actually performs better than Quark in CAST. Okay. So that's, so far I told about all the success of our method, but there are definitely limitations. And let me mention some of the limitations that the state-of-the-art program has. One is that it can so far fold less than 150 residues. I'm actually talking about basically the ab initio technique here, not the template base. Forget about iTaser. Here only, so we can only fold small protein, but in the last CASP experiment, actually, we were able to fold 3 proteins which were more than 150. But in general, we cannot fold accurately if the number of residues or amino acid is greater than 150. Another limitation actually currently is that we cannot fold beta protein with complicated topology. And what do you mean by that? So this is experimental structure of one of the target that was released in CAS 10. So here you can see it's a beta protein, and it has a huge complicated topology. So this is the N terminal where the protein starts, the blue. And this is the C terminal, this is the red one where the protein ends. And here you can see, although this C terminal is far away from N terminal, they are interacting. And if you see the schematic, so this is the N terminal, and the loop goes in this way, and then this C terminal actually interact with each other. Although they are sequence, sequence-wise, they are very far away, right? So we call it long range. Now, whenever we model this target using our ab initio model, it actually finds something like that. Okay, you can see this C terminal is actually far away from this N terminal. So there are 2 reasons for this limitation. One is that the current potential that we use, that actually cannot capture this long range interaction. Whenever I say long range, means like the sequence separation are far away. So we cannot capture this long range interaction with the current potential. That's one. The second is that, given the simulation time, it's very difficult, or the acceptance rate is pretty low for this this beta swap movement to come to here and have this interaction, because we cannot actually run this simulation forever. As I say, we only get 72 hours in CAS to submit our model, right? So if we run forever, probably we might be able to get that, but within a limited time, we have to get this model correct. So that's why with the current search engine and also the force field, this method failed to predict this type of method, predict the structure. Now, how do we solve that, or how we
Is that also the reason for the small proteins? Is too many fragments within the amount of time, or what?
There might be chance. If there is a complicated topology, there is a chance. Yes, because as I said, yeah, even if we have more than 150, but if the topology is reasonable, we might be able to find out, especially for alpha proteins. But for beta proteins, it gets difficult. Okay. So now, how to solve this issue, right? So one way to do that, if you think about it, if we know that the residue pairs which are far away from this, say N terminal, if we can know that which 2 residues are in close together, especially, or we call it contact, that information actually will be very helpful to guide our simulation. So if we know this information, that which 2 residues are in this, say beta strands are in contact or close together, or especially close together in 3 dimensions, this information, we can use this as a restraint in our Monte Carlo simulation, and that can be helpful for achieve this type of complicated topology. That's the hope. And that's why predicting contacts between the residues of the protein became a very hot topic recently. Okay, so in the next few slides, then I'm going to talk about how the contact. So let me give you first some basic information of what I meant about the residue contacts, or what is the definition. So say this is the beta strand I had, and this is another beta strand, and this is one of the residues in that beta strand. So this is the C alpha atom, the red, and this is the C alpha atom of that residue, and C beta is the blue, and this is the C beta. So if we find out that if the C alpha distance and C beta distance between these two residues is less than 8 angstrom, then we call it these two residues are in contact in the protein in the 3 dimension. Okay, so we actually classify this contact based on their sequence separation or range. So short range is like sequence separation 6 to 11, medium range 12 to 24, and long range greater than 24. What I mean by that, so this is the sequence, for instance. Okay, so this is the first residues. Now all the residues which are in green color, if they are in contact with the first residue or within 8 angstrom, then they are actually are in short range contact with the first one. Okay, and then because the sequence separation is between this one and from 7 to 13 is 6 to 11. Okay, now for the medium range, again for the first residue, if the residues in the blue are in contact or within 8 angstrom with 1, then they are in medium range contact. And if it is greater than 24, all the residues which are in contact with the first one, they are actually in long range contact with respect to 1. Okay, if we can get this long range contact accurate, that's actually that's golden. That's the overall hope. Okay, because if you remember in the previous slide, we want to look for the accurate long range contact. Okay, so as I said, it became very popular in this research field right now. So there are a lot of state-of-the-art programs which are trying to predict predict contacts before you run the simulation to fold your structure. So there are several types or several methods you can use. One is machine learning, where these are the methods actually using machine learning method. You can also use coevolution based method. So there are 4 programs right now. So coevolution based method is basically predict the contacts of the residues which mutate in a correlated fashion from the multiple sequence alignment. And there are some meta server which basically combines like this type of method, and then get the prediction. So in our lab, we developed a program called NEPCON, as I mentioned, and that's a meta program where we combine the contact prediction from this type of method. Oops, sorry. And then we get the contact prediction. So let me walk you over with our program. So NEPCON is basically neural network and bias classifier based contact prediction. So again, it starts from a query sequence, right? And we want to get the residues which are in contact. So first, what we do, we actually predict the contact prediction of this residue in this query using this program that I mentioned in the previous slide. So we have 8 programs that we first predict contact. And then we use a naive bias classifier to get the posterior probability of the residue pair in contact given the confidence score of the contact from each of these predicted programs. Okay, and then we all at the same time we also get some features for each of these position of the query and the neighboring position, like secondary structure, solvent accessibility, Shannon entropy, and so on. So overall, these and these we actually generate more than 700 features to train our neural network, and finally we get the contact prediction for the residue pairs. Okay, now again, as I mentioned, it's a prediction. So first of all, we have to see whether those predictions are accurate. If your prediction is completely wrong, then that information is you're going to feed in the simulation, and your structure will be completely off, right? So it's very important that you predict contact very accurately. So that's why here I'm going to show you that how NEPCON performs in terms of prediction accuracy. So we tested in 100 proteins where 98. 50 of them are easy, 48 of are hard. So I'm going to just show the 48 hard targets, the result that we got. Okay, and also I'm going to show about only the long range contacts. So whenever we get a contact prediction, the files look like this, the output. So this is the ith residues and jth residues which are in contact. And we also give a confidence score of each of these contacts. That means like how confident we are that these two residue pairs are in contact. And then we sort or rank based on their confidence score, like in the descending order of the score, okay? Now, we actually, like, as you see, as you go down, the confidence score getting low. That means if you are not confident, that means that prediction is not good, actually. So we only care about only top L by 5 contact or long range contact, where L is the length of the protein, okay? So, and then we predict the accuracy using this. So basically here, N correct is the number of correctly predicted contacts in this top L by 5, and NT is basically L by 5. So how many contacts are correct that we find out in the prediction. And here you can see along x-axis, these are the programs that we use in our pipeline, and this is our program, and this is the accuracy. And here you can see that our program, actually the accuracy is pretty high compared to other state-of-the-art programs. Yes?
Why do you take the top N predictions rather than using a probability threshold, like cutting off at, say, 95% confidence or something?
So as I said, you know, like this confidence score, like if it is pretty low, then you might get only, you know, one contact, right? So for instance, for this case, if you say 95% is the cutoff, you actually find out only one contact, right? So again, this is a prediction. So not all the confidence score is like, you know, probably like accurate, but the idea is the confidence score should also have correlation with the accuracy, okay? So that's why actually we have a kind of threshold, like, you know, say if you have, you're right, we actually use like say 50% or 20, well, 40% actually is like, you know, good if we have a threshold for using in the simulation, okay? But this is kind of standard in this community. Even in the CAS, they actually look for what is the top, what is the accurate prediction in the top L by 5, you know, and that's how also they assess in the contact prediction. It's kind of standard, but yeah, you're right, we should have cutoff as well, okay? So here, as I said, I'm just showing about the hard targets and long range, but we also did for short, medium, you know, and you can see this paper. But the overall idea is that, you know, the accuracy is pretty important, and we actually were able to do that. Now, we have to use this information in the simulation, right? That means we have to have a potential or develop a potential which uses contact. So we actually developed a potential which actually is in this form for contact, where this is actually like the distance and this is the energy. So this potential is centered at 8 angstrom because as I told you, like if the proteins are in 8 angstrom, especially they are in contact. So this actually centered from 8, and then 8 to 10 there is a strong force, and from 10 to here is basically, you can see the force is kind of weaker. So the idea is in the simulation, if we have a predicted contact, then it just pushes to this well and then try to bring closer, okay? And this Uij or this energy is the proportional to the confidence score of this the predicted contact as well. So using this contact potential, we actually merge this in the previous 11 energy term that I mentioned in the quark, and then the new pipeline looks like this. So it's this part you already saw for quark. Now we add this part using contact map. So first it goes to NEPCON, find the contact map, and then use this as a potential to guide our simulation, and then it's the same thing that we had before in the quark, okay? Now so this C-quark actually we used in CAS 12, that means which was like held in last year, 2016. So let me show you some results that we had using quark or C-quark in CAS 12. So here we have around 40 hard targets, I guess, or we call it free modeling targets. So this is actually showing the TN score of quark model if we don't use contact, that means the old quark. And here that if we use contact, which is used in like in CAS 12. So this is the C quark result. And here you can see that the TN score is pretty high, and even the average actually 3.3629, while if we use quark without contact, it's 0.2639. So there is a huge boost, you know, in terms of TN score whenever you use the contact. And for this specific target, if you see, for quark is only 0.25, while whenever we use 6 quark is approximately 0.68. So there is a huge improvement in terms of accuracy of this model. So this is the structure looks like. So this is the native structure, and this dashed line you can see this basically the contacted residue in the native structure. And this is the quark model that we got, which is pretty close, you can see. Now The reason we get again the good prediction here, because this is the contact map looks like usually whenever we have. So along x-axis is the residue index, and along y-axis is also the residue index. So each dot means like if there's any contact between these residue pairs. So this triangle is basically the contact that we see in the native, and this triangle actually that we have in the predicted model or predicted contact that we use in the simulation. So you can see that this like rectangle, these three actually kind of indicates that these contacts are basically for the beta strand, and this is basically for the alpha helix. And we are able to capture this contact using our contact prediction program, which really helped us to fold this to a correct structure. Okay, so here you can see how SeqWork actually performed really well in the CAPS-12, and this is the overall result that again top 10 groups which are in the CAPS-12 for 97 targets. And again you can see ZenServer actually performed better than SeqWork, and I told you the reason for that. And we are second, SeqWork is second, and like this is also our program. But here you can see the gap actually is decreasing day by day, and the reason is that they're also using contact. Okay, so if we would have not used contact in our Quark, probably would not be able to beat other guys. Okay, so here the question is, can contact really solve this problem of structure prediction program? And I think my understanding, my answer will be probably yes, because we actually tested 97 hard proteins where we use predicted contacts and the native contacts. Like we get the contact from the native structure, and this is the TME score looks like. So this is the TME score with native contacts and TME score with predicted contacts. And here you can see there is a huge like TME score improvement, 0.694, and if we use the native contact, while if we don't use native contact, just predicted contact is 0.439, and this is the p-value, which is significant. And you can see out of 97, most of them actually has greater than 0.5 TME score. And as I mentioned, if the TME score is greater than 0.5, that means it got the correct fold. So the idea is that if we can have a very accurate contact prediction, then probably we'll be able to solve this problem of the structure prediction. And that's why right now in this research area, people are like using different technique like deep learning or some other technique to improve the contact prediction accuracy. And if you can predict contact accurately, then you'll be able to predict structure accurately. That's the idea. Okay, so these are the web server that we have in our lab. This is the Quark, the old Quark. This is the NEPCON. That's the recently developed, and this is the SeqWork, which is combined with NEPCON and SeqWork. And since I have some time, let me go over the web server a little bit so that you can find out where to run jobs. So here again, let me show you the iTaser. I think you already know that, but I don't know whether you know, but you can also use iTaser. But let me show you the SeqWork. So here, this is the SeqWork web server. You can go to our ZenLab and then type SeqWork. So it starts from the sequence. So you can just, this is the kind of example input. So you can give your sequence, whatever you are interested in. But you have to register actually, and it's pretty straightforward. You can just, click here, register with your email, and just, give like confirm email, and you will receive a password. But this actually is freely available for only academic user. Okay, but for non-academic, there are some licensing issues. Okay, but anyway, so after you give your sequence and email and password, you will receive your results like in 2 to 3 days, we hope. So let me just show you an example output what you're gonna get whenever you submit any structure sequence. So this is the sequence that this output page looks like. So this is the sequence that you provided, and this is the secondary structure where for each position it provides you what the structure secondary structure looks like. So C for coil, H for alpha helix, and E for beta strand. So it provides you this information. It also provides you the solvent accessibility, which ranges from 0 to 9. So 9 basically means a highly exposed residue, while 0 is buried inside. And it also provides you the contact map that I showed in the slide. So again, this is the residue index, and the way it's color coded is the red is basically the highly confident, while the blue is there is no prediction. And also the good part is it provides you 5 models that it generates, and you can play with that, like look into that, and zoom in, zoom out, and you can also download this whole result. Okay, so this is the web server, and also since I have some time, let me also show you the NetCon. So okay, so this is the NetCon package. Web server looks like. Again, you just give your sequence. Here, you don't have to actually register. You can just give your email and password, or email and the ID or anything. It's going to give you a result. Okay, and you can also download. We have a downloadable package for these, so you can try these. Just download and just try these. Okay, so you have a lot of other programs that you can try. So with that, I would like to acknowledge my PI, Yang Zhang, who is the original developer of iTaser, and Dongshu is the developer for Quark, and Baoji is the developer of the first version of NetCon and CQuark. And I would like to also thank Cheng Xin, who is the developer for the CQuark web server. And these are the funding sources we have for this project. And thank you so much, and I would like to take any question if you have. Sure.
About, on average, how long does it take to run any of these softwares? Netcom, CQuark, iTaser?
It depends on the size of the proteins. Okay, so for CQuark, if you have like protein size of like say 150 residues to 200, it takes approximately 2 days or less than, sometimes less than 2 days. I think it's approximately 2 days. For NetCon, again, it depends on the size. But for 200 residues, it's going to be done in 10 to 12 hours, or sometimes less than that. It depends on the size. Okay. Yeah. So again, as I said, we have to do this like in CAS. We get only 72 hours, so we have to do this like very quickly. And these are all automated servers, so we have to do this like at least in 3 days.
So I think for like de novo prediction, we're still kind of restricted around 100, 120 residues for like good prediction. So after adding this contact prediction, how much have you pushed it by?
So as I mentioned, I don't have this slide here, but in last cast, we were able to predict 3 accurate models which had, I think, what was that maximum? The length was 300. So yeah, 300 residues we were able to reach. So yeah, but again, out of 97, only 3. Not out of 40 or 45. Those are hard targets. So there are 3. But again, it was like a huge success because So yeah, I mean, but that's with de novo. Any other question? Sure.
So if it takes this long for these predictions, what if you can get like compute time on, for example, the Anton, right? So where you...
So here, like our programs are not parallelized. So what is actually pseudo-parallelized. So what we do, we actually run 10 simulations in 10 different nodes. Okay. So now if you give me more nodes, then I'll be happy to take it. But our program is not like core-wise parallel. So even if we take Anton, it might be helpful.
It does, but I mean, for if you have like a small enough protein, like 100 residues, whatever, just fold it, right? Just let it run, and it can achieve like millisecond timescales. Then you can actually fold protein. Not for 300 residues, okay, but at least for 100 or 120 residues, you can still just run it for long enough.
Yeah, probably. So yeah, you're actually right in a sense because Anton might be more powerful. So we're actually using Exceed. Okay, so Exceed actually has more powerful resource than in our cluster, and we see that the same protein, whenever we run in Exceed, it's 1.5 times faster than our. So you're right. Like we can probably think of Anton, but
this is just comparing because I mean it's not that anyone can have Anton, right? These
sure. Yeah. We actually have to write a huge proposal to get this Exceed. We are approved this time. Actually, I think 10.5 millions SU, something like that, which is also pretty much decent, I guess. But yeah, that's a good idea. We can probably think to use Anthony if possible. But, yeah, it's expensive.
Yeah, it's just not feasible, but just trying to compare
Sure,
like
sure.
what other ways we can use.
Any other questions? All right, thank you.